Hello and welcome. I wanted to start by saying thank you for the opportunity to talk today. I'm going to talk for about 20 minutes on work that I did as part of my postdoc at the University of Oxford, which involved exploring the use of stem cell derived organoids to deepen our understanding of development and disease of the cerebellum. I'd like to thank several funders: Action for AT, Brash AT, Rose Trees, John Fell Foundations, and the Oxford Nuffield Medical Fellowship, who supported this work. I'm back in Australia now, and also would like to acknowledge the traditional custodians of the land, the Jagera and Turrbal people. I want to start by recognizing the colossal contribution that people like Yoshiki Sasai, Kiko Muguruma, and Mary Hatton have made to this area of research. I think without their pioneering work, we really would not be as far along as we are now. And so it's really through observing these studies of developmental biology that have allowed us to understand and to reproduce brain specification in a dish using stem cells. And so we know that development of the brain is controlled by these gene regulatory networks that drive the bordered gradients of morphogen patterns. There's a great example shown here, which is a depiction of the mouse embryo at E9.5. You can see that it's divided into segments: the forebrain, the midbrain, and the hindbrain. And so at the border of the mid and the hindbrains, we have the isthmic organizer, a really important signaling center, which is involved with development of the cerebellum and hindbrain. And so these critical early studies of developmental biology have taught us that sonic hedgehog and BMPs are involved with directing cell fate along the dorsoventral axis. And things like FGF2, FGF8, retinoic acid, and Wnt1 are involved with directing anterior-posterior pattern specification. And so I think there's a really great study that crystallizes this concept. So Martinez et al. Took recombinant FGF8 and they injected it into the telencephalon of a developing chick, showing that what you got was ectopic mid-hindbrain formation. So a cerebellum forming where a cerebellum would not normally form. And this tells us that there are these really critical upstream genes that are driving cell fate specification and the self-organizational capability. And so this really underlies the ability for us to induce tissue identity using stem cells and using organoids, which have become very popular in the last few years. So this is a timeline or a depiction of some of the organoid research that's happened in the last 100 years, showing that as far back as 1907, people were doing organoid research of some kind. So Henry Wilson showing that a dissociated sponge could reaggregate, self-organize, and form a new organism. And so really there are some arguments about what constitutes or defines an organism, but I think it's fair to say that an organoid is a construct of some kind that shows self-organization reminiscent of its parental organ or tissue. And so there've been some exciting advances with respect to a whole host of tissue types in the last 10 years. And part of this is thanks to the discovery of isolation and propagation techniques of human embryonic stem cells to come out of Jamie Thompson's lab, as well as the advent of iPS cells or reprogramming by Shinya Yamanaka. These technologies have made pluripotent stem cells accessible to a lot of labs worldwide. These are really powerful when combined with reprogramming of patient cells to generate these authentic avatars or models of affected tissue. Certainly there are limitations, including the purity and the functional maturity. And a lot of this is driving my current research and what I'll talk about. During development of the cerebellum, following the emergence of this isthmic organizer, development proceeds in two germinal zones, the rhombic lip and the ventricular zone, which give rise to the glutamatergic and GABAergic neurons respectively. These neurons migrate, proliferate, and expand to form the mature cerebellum. And in the mouse, over around 3 or 4 weeks, a small strip of tissue becomes this incredibly sophisticated structure, which is vital for our survival. Many of the processes that underlie development in the cerebellum are well understood in mice, but not so in humans. And so generation of stem cell derived tissues are a great opportunity to learn about the mechanisms driving development, albeit in a highly synthetic in vitro environment. So this is a diagram depicting the core methodology for producing mid-hindbrain tissue, which was published by Keiko Muguruza in 2015. This protocol really elegantly uses insulin and FGF to produce tissue with rhombomere 1 identity. And so this protocol starts with ES or iPS cells, which are dissociated and allowed to reaggregate in individual wells. They're induced in a growth factor-free chemically defined media with SB, which is a TGF-beta inhibitor, and also Y27632, which is apoptosis suppressor. It's this combination of FGF2 and insulin that have this mild caudalizing activity that give rise to these cerebellar precursors. And so as opposed to some of these older monolayer induction protocols, this method gives rise to tissue with spatial organization reminiscent of the first rhombomere or developing cerebellum. And so in our hands, this is what these organoids look like over the first few weeks of their life. And really importantly, we can detect cell types representative of these cerebellar precursors, including PAX6 and also calbindin, which presumptively marks Purkinje neurons. These cells transit through a stage where they mimic formation of a mid-hindbrain barrier, expressing lineage-specific transcription factors, including GBX2 and engrailed1. And this occurs in our hands with reasonable efficiency and reproducibility. On day 21, we see around 60% positive engrailed1 cells and around 45% positive GBX2 cells. We also see enrichment by messenger RNA of these hindbrain specific transcription factors at the expense of more anterior markers such as OTX2. And so day 21 in vitro approximates E9.5 in the mouse, and it marks a stage where the anterior posterior patterning gives way to this more mediolateral expansion of these subsequent germinal zones, that is the rhombic lip and the ventricular zone. And we can detect hallmarks of these events by looking at either kinetics or fixed checkpoints, which help us establish differentiation, maturity, and status. So here too, we can see chiral2, which marks the GABAergic ventricular zone progenitors, and MATH1, which marks the glutamatergic progenitors of the rhombic lip. And so what we see, depending on whether we look at protein or message, is reasonable consistency. These are 3 independent differentiation experiments. And so we think that on day 35 of culture now, we're around E10.5 in the mouse and maybe somewhere around 6 gestational weeks in human. And so the best indicator of a successful differentiation experiment is the appearance of this polarized neuroepithelial tissue, which expresses the surface marker chiral2. And so you can see here in this panel, there absolutely is some temporal and experimental variability. You can see cryosections through multiple organoids from 3 independent differentiation experiments. And you can get a feeling for what the variability looks like across and within experiments and within organoids. So one strategy that's been adopted is to use flow cytometry to purify out these precursors and culture dissociated cells with murine rhombic lip, which not everyone can do or has access to. We've chosen to avoid parts of this protocol for reasons which I'll explain later. Subsequent to seeing these ventricular precursor markers, we would expect to see the appearance of calbindin positive Purkinje neurons, which we do in fact see here. You can see labeled in 488 in this cryosection, and again in 488 in a whole mount of the organoid, the appearance of these neuronal cells that are immunoreactive for calbindin and have this bipolar morphology consistent with maturing Purkinje neurons. We also see biomessenger RNA upregulation of markers such as OLIG2, PTF1 alpha, FABP7, PAX6, and TAG1. And so now between this kind of day 35, day 50 window, we think we're about E14.5 in the mouse, or around 10 gestational weeks in human. This is really insight into a window of development that we wouldn't normally have. So these organoids do not survive indefinitely. And so we've looked into ways to improve their culture conditions to see if this increases the length of time we can keep them in culture. One of the popular methods adopted by many people working on cortical organoids is to encapsulate these organoids in matrigel. And matrigel contains ECM molecules such as laminin, collagen, and heparin sulfate proteoglycans, which may help promote differentiation through reproduction of signaling that happens at the basement membrane, which you can see depicted here. So evidence suggests that the basement membrane influences early patterning events at the mid-hindbrain. And one of the ways that it does this is orchestrating laminin-dependent basal constriction. So the ECM-enriched basement membrane of the cerebellum is located underneath the PL meningeal layer, where it supplies anchor points for the end feet of radial processes of ventricular zone precursors, as well as acting as a barrier to migrating neurons. These processes provide a scaffold which facilitates both cortical and cerebellar neuronal migration and correct layer formation. What we see after encapsulating these organoids is transformation. These organoids tend to become larger, flatter, and there's an outgrowth of neurons at the periphery of the tissue. We subsequently grow these organoids on transwell membranes at the air-liquid interface, which is a popular method that's been used for some time for growing cerebellar organotypic slices. And this, we think, helps improve with oxygen and nutrient diffusion into the tissue. Importantly, this helps us keep the organoids alive, intact, and in culture for upwards of 100 days. Apart from an increase in OLEG2, we see no obvious differences in lineage commitment after embedding these organoids. So the question that everyone in the field is trying to answer is, to what extent do these organoids really resemble the cerebellum or the cerebellar cortex? Do we observe organization reminiscent of the cerebellum? This is a really complex question to answer. Organoids don't necessarily follow the exact stereotyped patterning and formation of the brain. There's a breakdown of the scale and symmetry that orchestrates this event in vivo. And so these organoids do allow us to derive cell and tissue types that are produced via some of the same developmental programs that operate in vivo. They do allow us to study cell-to-cell interactions that would not normally be possible out of an animal. And so to explore this and serve as a pilot study for being able to use these organoids for modeling of disease, we decided to use single-cell sequencing to profile the composition of these organoids. Some of the questions that we wanted to explore were about inter and intra-organoid variability. We wanted to profile cells that we know about, as well as look for rare and unexpected cell types, as well as being able to benchmark the maturity of these neurons, which is really important. So we took 6 individual organoids following 90 days of culture. Each organoid was individually labeled with a hashtag oligonucleotide, ubiquitous cell surface marker. 3 of these organoids were embedded in matrigel, whereas 3 were in our standard conditions. And these were processed according to the standard Chromium 10x workflow. So really, the idea is to go from attempting to classify these organoids via immunohistochemistry towards higher dimensional space and looking at transcriptome of thousands of cells. This is a really big and ambitious process, as well as a big conceptual leap for us, and absolutely an ongoing process. So these are some sequencing summary stats. After QC, we ended up with 2 hash pools comprising around 1,600 single cells. And so this was processed according to an in-house Seurat version 2 pipeline that we developed. We've since moved to version 3. But the early version of analysis identified 7 populations. And so what you can see here are dot plots depicting some of the markers that were enriched in these populations. So you can see in panel F that we have enrichment in populations 0 and 6 of a lot of their canonical granule cell markers, such as NeuroD1, ZIC1, MEIS1, NCAM, DCX, and PAC6, with population 6 additionally expressing high levels of Ki67. In panel G, we can see enrichment in population 3 of canonical Bergmann glia markers, including FABB7, HOPX, GDF10, Vimentin, and SLC1A3. Panel H depicts a population where we see strong expression of Purkinje cell and ventricular zone markers. So we looked at the expression of some of these markers that had come out of the early analysis, and we cross-referenced these in the Allen Brain Atlas. And what we saw was that a handful of them were not restricted to one cerebellar neuronal subtype. For example, TCF7L2 and GAP43, both were expressed apparently in DCN and granule cells. And so this gave us a clue that we may actually be underclustering these cells at this resolution. And so what we noticed was that some of the genes that fell out of this analysis enriched as biomarkers had recently emerged from single cell sequencing of the murine cerebellum as marking multiple cell types. So glutamatergic DCN and granule cells have a developmental cell type of origin. Both emerge from the rhombic lip and express DCX as part of their subsequent migration through the nuclear transition zone or to the EGL. And so this led us to speculate that possibly at this resolution, we were underclustering. This is a really difficult issue to address because we have different abundances of cells at varying degrees of transcriptional complexity. And so we reran this clustering at a number of different resolutions. And you can see this animated GIF shows separation of cluster by increasing these resolutions, which is further depicted here by this cluster tree diagram, showing that we begin to approach saturation. And so eventually we could tease apart these populations and account for the majority of cell types by looking at these markers that were enriched and asking, do they make biological sense given what we know about the cerebellum? And so we've since moved to version 3 of the Seurat pipeline, in which we have cleaner separation of cell types and can account for the majority of cerebellar neuronal cells that we expect to see, as well as endothelial cells and an unknown cluster, which we've as yet been unable to place. Additionally, we're able to look at the composition makeup of each single organoid, showing that we can reliably induce these populations across our organoid cohort. And so to confirm our cluster identities, we used an available data set which comprised human cerebellar tissue from 9 to 22 post-conception weeks. We were able to integrate this data and project our human organoid data identity onto this integrated cerebellar tissue atlas object from Kathleen Millen's group, which you can see depicted here. So in panel C, we're looking at UMAP projections where each dot is a cell. The dots in gray are human cerebellar tissue, and the dots in blue are human organoid-derived cells. So you can see that we're projecting these cells in common transcriptional space, and we overlap with their tissue counterpart. Panel D shows the same plot color-coded to developmental age, so you can see a developmental progression of certain cell types. When we separate our batch-corrected average transcriptomes for each cluster by time, we can see that we can now more effectively classify our cell types with their in vivo counterparts. So we can see now that the organoid-derived Purkinje neurons cluster strongly with the 20 post-conception week Purkinje neurons from the tissue. Our organoid-derived ciliated cells, choroid plexus, endothelial cells, and roof plate populations all cluster most strongly with the 14 post-conception week choroid plexus tissue population. The granule cell populations within the organoid most closely cluster with the 21 and 14 post-conception week granule cell tissue cluster. Our Bergmann glia cluster most strongly resembled the 18 post-conception week Bergmann glia in the tissue. In agreement with this, principal component analysis with brainspan data showed clustering of pseudobulk organoid samples with mid to late prenatal samples, as well as showing a very strong enrichment for the cerebellum. And so once we were sure of our cell type assignation, we performed differential expression testing on each of the populations, showing that matrigel treatment elicited a distinct response in each cell type. This paper has just been accepted in scientific reports if you wish to read about this further. I think by far the most striking result was the fact that the matrigel treatment seemed to bias our organoids to have more representatives of the rhombic lip, that is, granule cells and glutamatergic deep cerebellar nuclei. And so this was apparent through marked upregulation of cell cycle and proliferation genes in these populations, which came out when we did gene ontology analysis. And so to depict this, we can see the same UMAP plots color-coded according to where these cells originate from, either the ventricular zone or rhombic lip. And so you can see a striking increase in the number of EGL and rhombic lip cell types in the matrigel treated organoids, which was borne out by chi-square testing at both clustering resolutions. And so while it's promising that matrigel embedded resulting in more rhombic lip cell types, this unfortunately comes at the expense of reproducibility, as the matrigel embedded organoids are much noisier in terms of composition relative to the unembedded group. So I think one of the really pressing questions is, what can we learn from these organoids about disease of the cerebellum? And this is a tricky issue because we know there are issues of heterogeneity and maturity. So we examined enrichment of disease-specific loci in specific cell types within our organoids. We examined genes causing Joubert syndrome, which is a recessive neurodevelopmental ciliopathy, and found significant positive enrichment in our ciliated cell population, which you can see depicted here. We examined the enrichment of genes that are associated with structural cerebellum malformations, cerebellar hyperplasia, and Dandy-Walker syndrome, showing positive enrichment in rhombic lip, granule cell precursor, and glutamatergic DCN populations. We examined genes associated with intellectual disability, showing positive representation in rhombic lip, granule cell precursors, and glutamatergic DCN. We found genes associated with autism spectrum disorder were also enriched in glutamatergic DCN. We didn't find any significant enrichment for genes associated with autosomal recessive and dominant ataxias that represent neurodegenerative disorders of the cerebellum. And this is consistent with the identified developmental stage of the organoids, in which most ataxia genes are not yet expressed. Thanks for listening. There have been a lot of people who've contributed to this project. Please tweet me or email me if you have questions. I look forward to the session. Thanks to the conference organizers.